Okay, so now we're going to take a look at the nervous tissue itself. Nervous tissue, let me enunciate here. All right, and try to bring this idea together, and then look at its function. So I've already mentioned neuron and neuroglia, right? I say elaborate on the neurons, which need to have a shape to allow many connections. So this is a network of cells, right, that really are trying to transmit information rapidly from one place in the body to another. All right. So the cell body, which is called the soma, is typical, but has lots again of mitochondria and rough endoplasmic reticulum, as we mentioned. Cell processes are key. The dendrites, as I mentioned, bring electrical change to the cell body, and the axons take it away. So this is unidirectional, one-way flow or change in electrical potential. The axonal hillock, which is right about here, is called the initial segment, and it's important. This is key to determining whether or not this neuron is going to respond to this neuron, because these are a network of responses or transmission from one part of the body to another. Myelin is made from glial cells. Here we're looking at the central nervous system because this is an oligodendrocyte, and we'll talk about this one in this video as well. If this was in the peripheral nervous system, these would be individual Schwann cells, all right? And so it's still called a myelin sheath, all right? But there are different cells in different parts of the nervous system that produce this myelin sheath, all right? Now, the myelin acts like insulation, all right? And it's imperative that it has gaps, because let's liken this to a wire that runs from any appliance, your computer or your washer, right, your coffee maker, to an outlet in the wall that is also transmitting electricity, which is basically flow of electrons, okay? So because you don't want that electricity to leak out and change the things outside of those wires, you wrap them with insulation, all right? And if there's a fray in the insulation, you replace the cord, right? Because that means that it's a fire hazard, right? Because that energy could initiate a fire, could be transmitted to and cause a fire. But in the axon, you're going to want these gaps, because here in the areas where myelin sheath is, there is no change possible. It acts just like insulation. But you do need a change to occur. You need a change here to eventually get down here. So these gaps allow for change to occur only at intervals or spaces, and therefore the transmission of the electrical change is going to be faster when myelin is present. All right, so types of neurons, again, the structural difference, just like we talked about structural differences in joints, right, with fibrous and cartilaginous and synovial. So there are trillions of neurons, many types, but most commonly distinguished by the number of processes, which are multipolar, bipolar, or unipolar, and they're shown here. All right. The multipolar are motor neurons, and that's what your model is. All right. So, and any neuron that's carrying information as a response to a muscle is a motor neuron, which is why we talked about that when we talked about muscle. All right. Most neurons in the central nervous system happen to be multipolar as well. It is very, very common, and multipolar because it has many dendrites. The bipolar are rare. They are generally found associated with the olfactory, your sense of smell, and the retina for your sense of sight. All right, but they are exclusively a sensory. They carry sensory information, sensation. All right, sorry, you're bipolar. So bipolar 1, 2 processes. Unipolar, so the cell body has really only one process. So it's pseudo unipolar because it splits. So it's one that comes off the cell body, but it splits almost immediately. Right? These are general sensory neurons. So there are dendrites that receive, right, and bring it to the cell body, and then axons that take it away. I mentioned here too, or your book says, that the cell bodies are always found in ganglia. So when we start looking at the spinal cord and we talk about the dorsal root ganglia, well, that ganglia is a collection of these unipolar neurons, cell bodies. And then outside that ganglion are these processes. right? So here's a cartoon for the neuroglia. And again, this is the neural glue. So they're support structures, and they can make neurotransmitters, growth factors, so they help with the generation of the cells. They help to keep ions balanced so that stimulation can occur, so electricity can be generated. They help insulate, as we already mentioned. They have some phagocytosis, so they can consume debris and damaged cells, and they also have some immune function to them. They really outnumber the neurons, quite a few more neuroglial cells. Most of these can divide, whereas your neurons don't, right? They are arrested in G0 of the cell cycle, right? There are 4 in the central nervous system and 2 in the peripheral. So astrocytes, microglia cells, oligodendrocytes, ependymal cells, these are part of the central nervous system, and you see them right here's 2 motor neurons being affected by these, and then the 2 in the peripheral are going to be the Schwann cells and the satellite cells. All right, so here looking at these specific types, these astrocytes, really cool, like star cells, right? And they kind of look like that. They're most noted for being part of what we call the blood-brain barrier, which is a very protective layering created by the cell processes of these neuroglia cells around the capillaries. So this adds extra filter or barrier, really, to the things that are in the blood, and it protects the brain, hence the blood-brain barrier. So it regulates passage of molecules from the blood to the brain and helps remove neurotransmitters. The ependymal cells, which have a different shape, they kind of look like these, right, simple cuboidal cells, but they have lots of cilia on them. And so cilia means we're moving something. Turns out the ependymal cells line areas in the ventricles, these spaces in the brain, and the ependymal cells participate with specialized capillaries in creating cerebral spinal fluid. So they then assist in the movement of this. And so they filter and absorb components of the blood, and they are really part, like the astrocytes, of the blood-brain barrier. But they basically line blood vessels found in capillary beds found in the ventricles and help to determine what cerebral spinal fluid composition will be. All right? Microglia are the cleanup, right? They are those phagocytes, and they have a different appearance to them. They look kind of like astrocytes, but that's a little bit deceptive. They're small, but they remove cell debris and bacteria from the central nervous system, and they control inflammatory responses, that's a very, very important function. All right? The oligodendrocytes, these are the cells that create the myelin sheath in the central nervous system. All right? And they look a little different than the Schwann cells. They actually, one cell tends to wrap around multiple axons, as you see here, whereas a Schwann cell, it's multiple Schwann cells around one axon. So the ratios are flipped. In the peripheral nervous system, we have satellite cells. These would be found in supporting the ganglia. So in that dorsal root ganglia, you're going to look at near the central nervous, near the spinal cord of the central nervous system. There will be satellite cells on that slide associated with those neurons. All right. They are similar to the astrocytes. They're kind of a barrier protective. All right. Schwann cells, they, again, only wrap around one segment of the axon, so they provide myelin. But like I said, here's one axon, and there's 4 Schwann cells here. So the ratio is flipped. There are more Schwann cells per axon, whereas there is more axons per oligodendrocytes when we looked at the previous cartoon. All right, both oligodendrocytes and Schwann cells are part of myelin sheath. It's just different locations. All right, so this is looking at the myelin sheaths. Again, we talked about this as insulation for the axons. So here is an actual image of this, and shows you this is a transmission electron micrograph. So many layers wrapped around. So collectively, the myelin sheath made up of a Schwann cell. So here's our endoneurium, which is a connective tissue around that, and the axon itself is contained within. Axolemma is a name for the plasma membrane of the axon, so it does, the Schwann cell is separate from the neuron itself, and axolemma is equivalent to like the sarcolemma, right? So myelin is a phospholipid protein coating and wraps around the axon. Said either a whole cell or a process. Whole cell if it's Schwann cells, process if it's the oligodendrocytes. It extends for only 1 or 2 millimeters, very, very short distance. Unmyelinated axons rest in indentations, so like this one, of the oligodendrocytes and Schwann cells, and there's not much myelin, this kind of, again, fatty substance, so this is gray matter. Myelinated sheaths, like you see here, is known as white matter. All right. Something else to understand about these myelin sheaths, it turns out that they can help direct repair of axons. Now, if the cell body is damaged, there isn't going to be any repair. But if a part of the cell is damaged, it can regrow, as I show you in this cartoon, through time. All right. So, and this regrowth is really guided by the presence of these Schwann cells. So say injured cell body will kill the neuron, but damaged peripheral axons can repair. And I say peripheral here because it turns out oligodendrocytes are not very good. They themselves do not proliferate. They're an exception to what we talked about for neuroglia cells. So regeneration is less likely if there's been central nervous system damage. But this is one of the reasons, again, why you really enclose that central nervous system. You surround it with bone and a protective barrier. So, again, after you damage the distal part, right, so here so if we damage site of injury, we'd have paralysis, right? There is no way to communicate between this neuron and this muscle. So if there's other motor units that weren't damaged, of course you can still get control of this muscle, but this pathway is gone, all right? So while this connection remains severed, there won't be any movement that occurs because no contraction. All right, but slowly, and again, I say 3 to 4 millimeters a day, we will see the Schwann cells divide, and as they divide, they will guide basically the axon back to contact. So you can regain some function if you have damage to an axon. All right, so function of nervous tissue itself. All right, so I say here, you know the players, you know the structures, you know the divisions of cells, and what they're doing and where they're found. How do we get all this communication? How do you get the things to do what you want them to, right? So nice cartoon here to connect the whole situation here. So in your hand, sensory, right, thermoreceptors. So we have, hot and cold water here. And the thermoreceptors if stimulated, can cause an electrical change. So we're starting off, there's our number one. So sensory stimuli. Amount of change, right, electrical change, depends on the strength of the stimulus, and this is what we call a graded potential. So the more change that occurs is going to be because there's more hot or more cold. All right? I say here, if you reach a threshold, which is enough change, you get an action potential, and we call that propagation. All right, so here's we're looking at this cartoon. Here's again our sensory, our thermoreceptors are free nerve endings found throughout our body, but here again we're looking at the hand, touching some water coming out of the shower head, right? All right, so here's the movement of the sensory. So sensory is the blue, so keep your eye on the blue, coming in. So here would be the cell bodies in a ganglia outside, right? So ganglia, part of the peripheral nervous system, outside the central nervous system. Here's central, here's central. All right, long, unipolar, right, neurons. So sensory taking it to the central nervous system and moves away from the cell body by an axon, and it could synapse here, right? So it could deliver this information to cell bodies here in the spinal cord, because this is all gray matter. All right, so here, action potential and sensory neuron, they're showing number 3. Sensory neurons enter the spinal cord and synapses with the brain. So we would go through, we'll call it an ascending tract, and come up to the brain. Now we're running out of time here, so I'm going to stop this here and carry it on in the next video.